cell cycle modeling is Combinatorial Therapeutics. So as a place to start, this is from systemsbiology.org. There is some similarities between information in the healthcare system and in a biological system from the point of view of health informatics and bioinformatics. Both of these disciplines are often fairly highly concerned with the flow of information. So organization of information, data handling, and importantly the interrelationships between these. So in the healthcare system, you've got relations between patients, doctors, governments. Another one I would put in here is medical researchers. I think that's very important to make sure that you're getting data flowing amongst all these groups. And to have effective communication between these groups is one of the main goals of health informatics as I see it. Bioinformatics, kind of by analogy, instead of being concerned with the individual entities, so with DNA or a given protein or RNA, it's often concerned with management of information and how these things interact. So the interaction, data handling systems, how to deal with large number of, let's say, RNA expression patterns or DNA or protein expression patterns, and how the machinery of the cell works. Instead of focusing on an individual cell or an individual protein within the cell, taking a look at the whole cell. Okay, so this is a very standard molecular biology. Oh, another thing I should mention, this talk I've sort of set it up for a multidisciplinary audience, so there will be aspects that some of you will have seen a lot, and some of you won't have seen at all. This is sort of very, very basic molecular biology. You've got DNA, makes RNA, makes protein, and then somehow that's expressed to a phenotype. And traditional molecular biology, biochemistry, in general follows a path that looks something like this. You have a hypothesis, you're often focusing on one gene or one protein, gaining some information about that. Once you have enough information, then you're trying to figure out how it fits into the bigger picture. So what are the relations between these individual entities? And then lastly, try to put this together and relate that to some effect on phenotype. So whether it's, if you're looking for a case in healthcare, you might be looking at a disease state, for example, what genes are involved in a given disease state, and you're trying to identify what those are. For bioinformatics, often what you're doing is you're using a more discovery-based research approach, where what you're doing, instead of looking at a given gene and working outwards, you're starting at the big picture and working inwards. So for something, what I'm showing here, this is a microarray, so you've got all these different bits of cDNA on here, and you put a sample on there and figured out essentially what messenger RNA, or by analogy complementary DNA, is present in your sample and at what abundance. So what you're doing in a discovery-based research approach is you're taking all this information. You might have over 10,000 bits of information on expression of individual genes. So then what you want to do is you figure out, you enumerate all those parts of a system, or what are changing. So you might end up with, in your given system, you're identifying 200 genes that have changed expression levels versus a control, for example. Or you could do the same thing with proteins. Then from that, try to identify patterns in the data and relationship between these various components. And then lastly, you're trying to relate these patterns to patterns of a given phenotype. So for example, let's say you're looking at identifying a given type of tumor. What microarray pattern corresponds to a given tumor type. Now, in practice, I think these things, people do separate them, but in reality, you've got a lot of interplay between the two. You're rarely looking at one gene in isolation. You're going this way as well. How does it fit into the bigger picture? And same thing here. If you find a few genes that are of interest, you'll go this way and focus in on them a little bit more. So it's not like these things are separate. They do eventually come together to provide an effective description of the system that you're studying. So this is kind of an optimistic view of genomics and proteomics. You got your traditional geneticist or biochemist here fishing for a given gene, for example, and then genomics and proteomics a little bit more of a brute force approach. Nice cartoon. The thing is this realistic? Often, you need kind of both these approaches. One of the biggest drawbacks of something like this, yes, you get a lot of information, but trying to figure out what it means is the real challenge. You have 200 genes, maybe 3 or 4 might be important. How do you figure out which ones they are? So that's where this comes in. You're not quite sure what you're getting. Okay, so applications of bioinformatics in the health sciences. One thing I should mention, bioinformatics, genomics, proteomics, still it's essentially a recent field. So definitely plays a big part in research, but transferring research into an actual application in the health sciences takes a little bit more time. So most of the stuff that's been done is kind of testing the waters. What can we find out? What can we figure out about what these tools, what problems can these tools be applied to, and are they better than existing diagnostic methods? So one example, genetic profiling. Now this is already, can be done to a certain degree. If you want to be screened for certain genetic predispositions, you can do that. You can be tested for, let's say, a cystic fibrosis gene, for example. What bioinformatics can do is it does that same thing, except now you might be able to do it on a larger scale. Instead of testing for a specific gene, maybe you can test for 20 or 40 or 1,000. So now this actually gets into an interesting territory, because predisposition, that's an interesting term. Insurance companies will not insure pre-existing conditions. So if you go in and they have this information, it says, okay, you've got this particular gene, which means you're going to get some sort of terminal disease at the age of 40. They might say, well, that's a pre-existing condition. We don't want to deal with that. So therefore, you're not going to get insured. So now it's not at that point yet, but it's something to consider. Any technology, any new technology, have to think about what the implications, what societal implications there are for these things. So that's just a bit of an aside. That's kind of an interesting area, I thought. Okay, now from a more practical point of view, prognosis and treatment. Genetic predispositions are one thing. Another way of dealing with information is assessing a current cellular state, whether you're doing it through looking at messenger RNA, what genes are being expressed in the cell, or proteomics, what proteins are being expressed in the cell, at what level, and at what type. Essentially, what kind of isoform of a given protein. So some things that are possible, and people have started researching this, prognosis and treatment of disease. In some cases, you may be able to predict the response to a given treatment. So some cancers are known not to respond to certain treatments. If we can identify what this cancer is in advance, then we know, okay, option A is not the best one to choose. We can go to option B. Now again, this hasn't been done in practice, but a very interesting area, and they've already shown that you can definitely identify various different tumor types using bioinformatics technologies. So something related to that, you might be able to tailor the treatment according to a given tissue subtype. So what is going to be the best possible treatment for this observed cellular state? Another important aspect of this, here's where you know where the problem is, and this is sort of fits with current knowledge. Often you're dealing with a biopsy, you already know there's a problem. One of the real promises of something like bioinformatics is early detection. In theory, would you be able to go into the doctor's office, take a blood sample, and they either analyze protein content, peptide content, messenger RNA, and say, okay, you've got something going wrong with your liver, or you've got early stages of cancer, something like that. So early detection. Now this is something that remains unproven. People are getting there. They're able to take a look at a blood sample and correlate certain patterns with certain disease states, but it's not at the point yet where it could actually be useful in diagnosis. But potentially, it might be. And this would be one of the real advantages of using these approaches in the healthcare system. Something else you can do, often, if we remember the fisherman, you're fishing for new things. You might be able to identify a protein that's correlated with a given cellular state and not know what that protein is or what that gene is. So in doing so, what you might be able to do is to say, okay, well, find out what this is. It might be a useful drug target. Something else that's related to that and related to combinatorial therapeutics is the idea of multi-factor diseases. Now, if you have a genetic disease where you know there's a given cause or causes that all sort of lead to the same cellular state, cellular or disease state, that's fine. But in many cases, you're going to have not just a single cause, you're going to have interactions between a number of causes. Alzheimer's would probably be an example where you can get genetic predispositions, but actual start of the disease is going to be multiple factors that actually cause it. Okay, so that's some of the promises of bioinformatics. Now, what are some of the problems? One is this: highly dimensional data sets. You have a lot of measurements, and often, you're only going to be comparing a relatively small number of treatments. 2 is possible, maybe 5, maybe 10, but whatever this number is, it's a lot less than this. So it's very hard to, you can find patterns in this, but they might not be particularly meaningful. They might just be an artifact of the fact that you've got, only relatively few samples and a huge number of measurements. So you might get, from a computational point of view, let's say you've got a pattern recognition tool of some sort, support vector machine or whatever. It can analyze perfectly the given data set that you've got, but it's only good for that data set. You can't extrapolate outside. So that's a potential problem. From a biological perspective, this is a big problem. What does it all mean? So analyzing microarray data or proteomics data, it's very, very hard to sort out. You've got 200 genes that have changed expression patterns. Andrea knows about this. Trying to figure out which ones are important and which ones aren't is a major challenge. Also, you can have a black box approach where you can have a system that will recognize a given state, but you really have no idea what's happening in the biology. Just saying, okay, when this given set of genes goes up, you know that that represents this state. But at a biochemical level, you have no idea what's happening. This is also something to be considered, cause and effect. So often if you're looking at a given disease state or cellular state, you want to know what's caused that. Like what change has initiated all these other changes within the cell. So varying different levels of proteins. So often you're going to get, let's say, we do 2D gels in the lab. We'll get 100, 150 proteins recognized as changing expression levels. Whoop. 2. Okay. Yeah. So you get a large number that are changing expression levels. There might have been only a relatively small number that have caused that change, and the rest are just secondary effects. So you're just taking a snapshot, and it's really hard to figure out which ones are which. Data management, anything in bioinformatics tends to involve a large amount of data. There are systems in place for dealing with this, but conceptually it's a little bit harder to grasp in many cases. From a healthcare perspective, often, or at least right now, it's not necessarily practical. You can't regularly do microarray analysis of samples from people because you're looking at probably 200 US per test, and you're not even sure what you're looking for. So that right now may be a limit, maybe an obstacle. And right now also, the equipment used for a lot of this analysis is very expensive. And consumables are as well. Okay, so I wanted to talk about a couple of examples from the recent literature. One, diagnosis and treatment of breast cancer. Another one, approaches for biomarkers of disease. Identification of drug targets. I'm not going to talk about this too much, but it's just something to keep in mind that when you are identifying these differentially expressed genes and proteins, some of them may be viable drug targets. So it might be useful for pharmaceutical development. And then lastly, this is some of the stuff that I'm doing in collaboration with Bernie Dunker and Brian Ingalls in University of Waterloo. If you can model a cell cycle, if you can come up with a good description of a cellular system and figure out, how you might be able to control that to a certain degree, then that might have implications for medical applications. So, example, if you can control cell division in a simple system, maybe you can control cell division in a more complicated system, like a human cancer. Now, that's still a ways off, but in theory, it's possible. So this is your standard microarray experiment. You've got some sort of reference sample. You've got some kind of RNA that's been extracted from a cell sample or a tumor or something of interest. Then what you do, you take these, amplify it, get cDNA, put it on a microarray, and each one of these spots, what it represents is a complementary cDNA which can bind either of these guys. Now, the only difference between RNA, let's say for a given RNA, you've got one version over here, you've got the same version over here. The only difference is they've got these different fluorescent tags on them. So when you hybridize them on the array, you end up with something that looks like this. And spots that are yellow, more or less, are an equal mixture of these. Something that is red has much more cDNA from this sample. Something that's green, much more from this one. So what you can do is use this, and you know what each one of these spots are. So here you've got ERBB2, which is one gene that's involved in breast cancer. So given that information, also a little bit more background here. For your typical, or for at least one type, so Stanford Microarray, human microarray, what you've got is you've got over 20,000 expressed sequence tags, so these little bits of complementary DNA represented. This is an important part. So what you've got is about 40% of these things, as of this paper, 40% of them, it's reasonably well known what they do. 10%, a little bit of information. About half, very little information, other than it's a gene that's expressed in the human tissue. So that's it. So what that means is that half of those spots, even if they change in regulation by a large amount, you have no idea, what is actually happening at the cellular level. Okay. Yeah. Another thing I wanted to mention: you've got standard reference RNAs, so you can make comparisons to a standard. You don't have to be just comparing pairs all the time. You can work. What they've done is they've taken 10 human cell lines, pooled the cDNAs, and you can get that as a standard reference for comparison. Okay, so a little bit about the data analysis. You've got all these spots. How do you handle the data? How do you make sense of it? So for each spot, let's say you've got 20,000, for example, you can represent it as 3 bits of information. So what the cDNA is, which sample, whether it's your treated or your control, and how big of a change you get in expression. So changes in these expression ratios can be represented on a color scale. So black is essentially the same, unchanged. Red is induced in your test sample. Green is repressed. So this is all important stuff for the next slide, so it'll make a little bit more sense. Then what you can do with all these data points is you can cluster them by similarity between samples if you have multiple samples, and you can cluster them by expression similarity. So expression similarity, you've got 2 genes that in various samples they're always correlated. They're going up and down at the same time. Same thing, 2 samples, very, very similar. You can put those together. So in this paper, what they did was they looked at 115 breast tumor tissues and 7 non-malignant tissues and see if they could figure out not only which ones are non-malignant and which ones are tumors, but how these various tumors can be divided up. So you end up with something that looks like this. So this is the whole data set. Each one of these little spots in here So vertically, each column is a sample, and each row is a given gene or a given spot on your microarray. So from this data set, what they did is they did a cluster analysis. So they tried to group all the genes that behave similarly together and all the samples that behave similarly together. And from all that data, they said, okay, well, we recognize these particular genes as being the most important or most indicative of given cellular states, and then put those here. So what you end up with is you've got five different gene types, and they've also could detect various forms of cancer. Here's your normal tissues. Then you've got a basal subtype, ERBB2 expressed at a fairly high level, then luminal subtype A, luminal subtype B, and then these gray ones, they didn't really trust where they go, so they just sort of said, okay, we're not going to classify them, which is perfectly reasonable. Okay, so what they've done from these tumor samples is they can tell various types from the gene expression patterns what class they can fall into. So from a health perspective, very nice, but why is this important? If you take a look at survival rates for these different types, here's the luminal type A. Survival is somewhere between 80 and 90%, at least 85. Some of these other types, survival extremely poor. So this could be highly relevant to treatment plans, because you know whatever treatment, assuming all these guys had the same treatment, then these ones are not responding to that given treatment. So now this is not used in the health sciences right now, and you can get similar information possibly by someone who is very good at histology. They might be able to distinguish various different types, or possibly they might not. For this particular case, what they've divided up into known types of cancers, extrapolate from known information, but it is possible that they can recognize, and there have been cases where they've been able to split tissue types into two new groups and found out they have very different properties and very different responses to treatment. Okay, so that's one example. This is another one, and this is still more or less at the theoretical stage. But what you could do is it's possible you might have biomarker amplification and harvesting by carrier molecules in the blood. So say this is your circulation in here. This just represents the body, more or less, various cellular compartments. And what you're getting, let's say you've got a cell here that has some sort of problem, some sort of given disease state. And what might happen is some of them are going to break down, and you're going to get biomarker proteins released. You're also going to get, let's say for a tumor, you're going to get necrosis. You're going to get some of these broken down. You get proteinases, and what will eventually happen is you're going to get these little fragments of proteins that are going to go into the circulation, and they're going to bind to serum albumin or other proteins that are part of circulation. And then you've got all these possible markers for various disease states. Then from that, what you can do, you've got your protein carrier, probably albumin, these little peptides stuck to it. You can desorb them. This is just one possible technique. Essentially, it's a way of looking at what peptides are within the bloodstream. You release these peptides and then analyze them by mass spec, and then you'll get these various tags. You can identify what all these peptides are, or you might be able to identify what these various peptides are, and then from that figure out where they're from in the body and what cellular state they represent, healthy, disease, so forth. Now again, this has not been put into practice, but some preliminary work has been done on this. Now, something else that's interesting about this is, and this gets back to the whole discovery-based versus hypothesis-driven research. It's possible to just take, this is a mass spectrum, various peptides, so you've got the mass down here. You get a whole bunch of peaks for various different cell types. So this would be normal cells. This is from brain tumor biopsies. Grade 2, astrocytoma. Grade 3, astrocytoma. Glioblastoma. So 3 different types of cancer and normal cell. So without knowing what all these things are, you can still distinguish, you can classify these various groups. Now, this can make biologists a little bit nervous, rightfully so. You don't know what you're looking at, other than you can say, okay, this pattern represents this cellular state. That's all we know about it. But if it can be reliably used for diagnosis, that's fine. Maybe figure out what some of these things are later. Because what happens is when you put this into a classifying algorithm, so like this clustering algorithm, similar to what was used in the microarray, you can classify it into these grade 2, grade 3, grade 4, and non-tumor cells. In this particular example, they have 100% accuracy, well, with that one possible outlier. So this has the potential where you can actually do diagnosis without really knowing what you're looking at. Potentially useful, but again, the big question goes back, okay, this worked very well for this particular data set. Can you extrapolate from this data set to other scenarios? Okay. Now I wanted to talk a little bit. This is some of the projects we've started in the lab. Fairly preliminary work. I haven't been here that long, but given the opportunity, I wanted to share a little bit of it. So one of the main techniques we're using is proteomics, using a variation on 2D electrophoresis. For people who aren't familiar, essentially what this is just a way of taking a mixture, a complicated mixture of proteins, several hundred, maybe a few thousand proteins, and separating them out according to their inherent biochemical properties. So for 2D gel electrophoresis, what you're doing, separate first dimension by their charge. Each protein has an inherent charge at a given pH. So what you can do is if you put these proteins in an immobilized pH gradient, put an electric field across that, they're going to migrate to the point at which they have 0 net charge. So, and that's termed isoelectric focusing. Second dimension is standard polyacrylamide gel electrophoresis. You take your proteins that you've separated by charge in one dimension, put them on top of an SDS page gel, and what happens is they're associated with sodium duodecyl sulfate, which gives them a uniform charge. You put them in an electric field, and they'll migrate through a gel according to their molecular weight. So, let me skip ahead here. End up with something that might look like that, a whole bunch of spots. Okay. So, a fairly new twist on this. One of the problems with 2D page, it's great. You can separate out all these proteins, but quantification is a little bit of a problem, and repeatability is a little bit of a problem. So it would be nice if you could do something like a microarray, where you've got your 2 samples, you can compare them directly, you know this spot exactly matches up with this spot, and you can quantify them. So for differential gel electrophoresis, this is a way where you can do that. So instead of running everything out on your gel and then having some sort of staining protocol, what you can do instead is much like a microarray. You start off with your 2 samples. Before you put them on the gel, you label them with two different dyes. So now these are particular in that they have the same charge and the same mass, so they behave identically on the gel, which is a very important consideration, and this is probably what slowed down this procedure from becoming commercially available. But now they've got it to work, so what you can do is you can mix these 2 samples, run them out like on a normal gel, and then if you have a scanner that can pick up selectively these 2 different dyes, you can get 2 different images. Then you take those 2, you can overlay them, and what will happen, now you can't really see it very clearly here, but you've got all these spots overlaid, and just like with a microarray, these slight variations in color can be correlated directly to variations in protein abundance and quantity. And then there's nice software that goes along with it. So you can, this would be a representation of these various gels. Now what's been highlighted here is proteins that have been selectively upregulated or downregulated. So all the green ones saying, okay, all these ones really haven't changed all that much. The ones that are statistically significantly different, here the red ones, I believe, are downregulated, blue ones upregulated. You can quantify those and come up with the differential expression between each given protein spot. And you can also make pictures that look like this. This represents a spot on a gel in 3D, same spot in the other version. And you can show, it shows that, okay, it's present in this sample, missing in this sample. So it might be an important protein for whatever system you're studying. Okay, so the advantages of this, the big ones, you can run more than one sample in the same gel. It really allows ready comparisons between the two. You can match these spots versus an internal standard. That's an automated procedure. This is also important. Not only are you getting fairly accurate differential quantitation, if you have highly reproducible results, you might be able to do this down to about 10% differences. Whether or not that's biologically significant, hard to say, but you can quantify at that level. And importantly, you can come up with statistical significance. You can run multiple gels and say, okay, you know, sure, this is upregulated by 50%. Is that statistically valid or not? And how you can do this is by using multiple gels versus the same internal standard. They can all be correlated to the same internal standard. And the end result is this. You get a large reduction in the variation. So what used to be a problem in comparing these gels and figuring out, you know, matching up which spots are what and the amount of variation between the two, now you've got a much more accurate measure of that. So it becomes similar to microarrays. So now limitations of this is not perfect. You don't get any information on subcellular localizations unless that information was available beforehand. If you're only looking at a nucleus, you can say this protein is likely from the nucleus. Otherwise, you don't know where it's from. Low abundance proteins, difficult to pick up, but you might get around this by prefractionation of some sort. In most cases, you're just comparing snapshots, so you don't have kinetic information unless you run like a time course. And this also is important. The functional unit of a protein is often composed of these multiple subunits. In this process, you're splitting them all up, so you have various isolated subunits, so you lose that information, which may or may not be important. Okay, so I wanted to relate this to applications in healthcare. So one example, you might be able to use these protein expression patterns to distinguish between various cancer types. So similar to what you had with microarrays. You might be able to determine origin of metastatic tumors. So you have this metastatic tumor, what original tissue type is it from? An example using DIG, using the same technology, classification of lung cancer subtypes. So this is where you get this gel. Now this is an image from a paper, so they lose all the nice colors, so it's just a black and white. But you can see is that they've identified quite a variety of upregulated, so these triangles are upregulated in this particular lung cancer, which is a small cell carcinoma, and all the circled proteins are downregulated. So just given this, without even knowing what these things are, you can say, okay, this is representative of small cell carcinoma. You know what tissue, what type of cancer it is. Same thing, doing classification studies on this. Here you've got a principal components analysis. Here you've got a cluster analysis. You've got 3 different types of lung cancers. Small cell lung carcinoma, which are these guys. Squamous cell carcinoma, mostly these. And adenocarcinoma, which are the green ones. And you can see that it didn't do a perfect job, but this is still reasonable. You know, it separated out probably about 90% or more were correctly classified. There's just these three outliers. And then principal components analysis shows a similar pattern where you've got separation. Now this is a 2D representation, something that's 3-dimensional, but you can still essentially see you've got these differences. Now, relating the discovery science to more the hypothesis-driven science, what are these various proteins? you can see that they identified a fair number, but then you also get some of these here. So 5 down there, a couple other ones right there that haven't been identified but are still representative of a given cellular state. Peroxyredoxin was, for example, upregulated. And what you can do is you can take this information and try to relate it to what you know about the biochemistry of these various cell types. Okay, so something that we've done in the lab is we started looking at differential expression of proteins involved in cellular senescence. So this is in collaboration with Gerardo Ferber of the University of Montreal, and he's looking at cellular aging. And what he does is he has these fibroblasts, human fibroblast cell lines, and he can induce a state of cellular senescence using RAS oncogene. So this is senescence-like. It's not exactly the same, but it's a cellular model. Pretty reasonable. And then what we're doing is we're comparing the control and induced cells, senescence-induced, using this differential gel electrophoresis. And then from that, the proteins that are differentially expressed, we're taking a look at them by mass spec. So we can make cool pictures like this. So what this is your standard 2D gel. Because we're using fluorescence as a means to measure this, this is why we get this inverted color scheme. But what you can see here, very, very similar to microarrays, what you can get is you can pick out these green ones, these red ones, where the green represents, in this case, something that's upregulated in the senescent cell, and red represents something that's downregulated. Green and red are kind of arbitrary. You just keep track of what's what. So from that, you can see that there are quite a large number of changes. Like there's little guys in here, there's some green ones in through here. But the ones that we've identified are the ones that are circled. So you can see there's one here that definitely changed expression patterns. Same with this one, that one, hard to say. So then from that, we can excise these, cut them out of the gel, analyze them by mass spec. So this is just sample mass spec data. What we've got from this particular fragmentation pattern. So we take peptide and then put it through the mass spec. It gets fragmented, and you can see here it's split at, or odds are it's going to be split along these peptide bonds, and you get this characteristic fragmentation pattern, which corresponds to this amino acid sequence. And if we have a couple of these that are both pointing at the same protein, we can say, okay, yeah, that particular spot is this disulfide isomerase ER60. So now in doing this, we're still left with the challenge, okay, well, what does this mean? Which is sort of where we're at now. We have to try to sort this out. But we did get some interesting results. So we've got here are session numbers for these various proteins we've identified, ratios between the senescent cells and the normal cells. Now here, these ones you can see they really haven't changed much. And these are things like lectin, lectin-like protein, filament protein. Some microtubules haven't changed a lot, but then there's some other ones that have changed quite significantly. For these ones, cytoskeletal proteins, these have both been upregulated significantly in the senescent cells. And you're also getting some other ones that are quite sharply downregulated. This one is kind of interesting. Collagen is about 18-fold downregulated in the senescent cell. Yeah. Well, a standard experiment, what you'd be doing is you'd be running probably 3 or 4 gels. So you'd be running replicates. And then when you do the analysis, what they're going to do is they'll give you these numbers, but it'll also give you a corresponding p-value. So is this statistically significant? What is the variation of these proteins between the samples? So you can sort those out. Odds are, like for something to be looked at in detail, you could say, okay, we want it to be significant p-value, so p less than 0.01 or 0.05, and probably also 1.5-fold or 2-fold induction. So you try to balance these things out. In practice, what you end up doing is you start at the most significant and you work your way down. So, yeah, so you can deal with that essentially, and you do get reasonably repeatable results. Sorry. Well, ideally what you should be doing, now we're just using cell lines here, but you would want them to be true replicates. So, you know, different cell lines, different beakers, you treat them individually until you run the gel. So you don't want to take the same sample, split it into 3. That would give you a measure of the variation of this particular piece of equipment, but it's going to be a lot less than biological variation. So, yeah.
My question is, how reliable is that number? I realize that you can use the evaluate the
Yeah. Yeah.
But the problem I see is concentration. I think in terms of learning, with a 2.5 microgram, 2.5.
Yeah, that can be a possibility, like if you do get saturation. But one thing that you can do with this, because you're using fluorescent dyes, if you get a spot that's saturated and it's an important one, you can probably just rescan the gel at a lower intensity and it'll drop it down. So you can get you can get a reliable measure of the ratio essentially. And it's quite repeatable and it's linear over 4 or 5 orders of magnitude, I think, something like that. Yeah, so it's fairly good. So yeah, you can trust these, although like most things you have to make sure that the conditions are right, that you're not saturating and so forth. And you can also get error estimates on these numbers as well. I haven't shown those here. Okay, so one other thing that I wanted to point out about this list, again this is preliminary, but this one was quite interesting. Transgelin, this is known to be linked to replicative senescence. But what's interesting is that link was found through RNA expression patterns. So microarray, a type of approach. And protein data and messenger RNA data don't always correlate. And this is one example where it doesn't. So in replicative senescence, you're getting the RNA upregulated, but the actual amount of protein, surprisingly, is less. So we're not sure what that means yet, but it's a difference. Okay. Back to that in a second. This goes back to the whole idea of systems biology and trying to sort out these things. The next thing I want to talk about is some of the cell cycle modeling and what we're going to do. We've only started this project relatively recently. So this is one way of looking at interaction data. Probably not the most useful. But what you can do is you can actually make use of the inherent organization of biology. What you get, much like, you know, you've got an organism, can be divided into organs within that, cell types within given organs, keep going down. You're going to get organizational units where you've got subcellular components, you've got pathways within those, individual proteins and genes within those pathways, and then connections between the two. So from this fairly simple model, you can build things that are actually can be interpreted. They start looking a lot like circuit diagrams, actually. And if you know what you're looking at, you can come up with something like this, which is a little bit better than just that whole mess of lines. Okay. The question was, what approach do you use to do this sub-clustering? As far as I know, now this one isn't my research. This is a very complicated pathway that's involved in sea urchin development. I think it's done by hand. So Bernie might know about that. Yeah, okay. Yeah, I would think that this is done by hand where people are trying to relate or show the interrelations of all these various genes and proteins, and also dividing them up into these various subunits. So, although it's relatively complicated, the idea is for this eventually to be a reasonably accurate depiction of your biological system. Yeah. Yep. I know. Yeah, there's I know one guy at the Weizmann Institute in Israel. He's actually taking a look at these expression. Oriolon, yeah. Yeah, and he's essentially that's what he's doing. He's saying, okay, well here's these interrelations between genes. How do they relate to circuit diagrams? So this is a feed-forward loop, this is a feedback loop, this is a gate array. And so it actually lines up with electronics or logic, or can. So yeah, so it's an interesting area for sure. And I think it's, as far as I know, it's currently underdeveloped. So if you want something interesting to do, it might be an interesting area. So what are we doing? Now I wanted to relate, we're looking at cell cycle modeling in yeast. I kind of want to relate this to health informatics, so you can think of these as tiny little patients. So, yeah, so in cell cycle modeling, what we're doing, taking a look at genes, proteins, and interactions involved right at the initiation of DNA replication. So we're not trying to figure out everything to go on, everything at the beginning. We're saying, okay, this small subcomponent, let's try to figure out what are all the proteins that are involved in that transition. Just saying, okay, start DNA replication. So we're using a couple techniques here. One, this is my part of it, using DIG to isolate, quantify, and identify these differentially expressed proteins and protein isoforms. Now, one of the interesting bits about this system, about DIG, is you can actually pick up different things like phosphorylation states. So you can pick up different isoforms, which may be very important in cell signaling. Communications between proteins very often are mediated by phosphorylation and dephosphorylation. So we can actually pick those things up. Then once we get that information, then ideally what we'll be doing is modeling the protein levels and the interactions as a set of differential equations. So some work has been done. A nice thing about this is you don't have to try to come up with these complicated pathways entirely on your own. You can use existing information and build on it, much like most of science. So we have this model, and then what we want to do is study it and refine it. So what we can do, ideally, is we want to use a cell cycle model to predict the effect of perturbations on this system. So we come up with a model. Genetically, we can play around with expression levels of given proteins. So let's say what happens if we remove this protein or we overexpress this protein. And then we can say, okay, this is how we expect our cell cycle model to work. And then we can go back and say, okay, well, is that what is observed? And if it is, great. If not, then we have to go back and figure out why not and try to refine the model. So one addition that we've made to this is one of these pathway databases from KEG, Kyoto Encyclopedia of Genes and Genomes, which is an interesting source for these things. So you can see fairly complicated, but what we're interested in is the transition, so these proteins in here. Now, this is sort of known information. Doesn't mean that it's a complete representation, it's just sort of what's known about this system. And one of the things that was hypothesized by Bernie Dunker in the Department of Biology is that this important protein, DBF4 here, interacts with HCT1, another protein that really doesn't, you don't see a connection between these two here. So can we use this extra bit of information to further refine the model? So in using our DIG, I've gone back to the black and white here, what we're comparing is an HCT1 knockout. This is just preliminary work, but essentially we've got HCT1 knockout, so we can do the DIG comparison between a wild type and this knockout of the yeast protein, of the version of the yeast, and we can see, okay, yeah, we get this particular protein has disappeared. We have to confirm this by mass spec yet, but we believe it's HCT1. These other big spikes are bits of dust, which show up very nicely as well. They fluoresce quite well. But they can be pretty readily distinguished between the proteins. So you can see this one is still present, and this one's missing. And then from that, you take that information and say, okay, well, how well does that fit with our differential equations? What's going to happen to these various protein levels when we include this in the model? So we've got HCT1, which was modeled already within the Chen-Tyson-Novick model and also CDC20, which is this blue one. So given that, when we include this in the model, what happens to DBF4? And we predict this is the pattern of expression we're going to get. So now the next thing that we have to do is actually see if this is the case. If yes, then we keep this in the model. If not, then we have to come up with some other way of figuring out the levels of DBF4. Maybe there's something else that's involved. Maybe this link that we've hypothesized isn't actually accurate. So this is what we're doing right now. This is kind of in progress. And in general, as we're doing this cell cycle modeling, we're doing a bit of a cycle ourselves. And again, this is myself, Bernie Dunker, Brian Ingalls in the Department of Math. And what we're doing, taking the information from DIG about the differentially expressed proteins and protein isoforms, refining this model, going back, saying, okay, well, given that we hypothesize this new model, let's actually tinker with the yeast genetics and see if we can predict the effects, and then feed that back in, measure the system again, and keep going. And the idea of doing this, essentially what we want to do is we want an accurate description of the gene-protein network involved in the initiation of DNA replication. So what are all the representative proteins? How do they interact? Can we predict what they're going to do? That's really the crux of this, is what we want is a predictive system. So if we can do that, then maybe, eventually, many years in the future, we can do the same sort of thing for human cell lines. Say, okay, well, can we predict how they're going to behave given this given perturbation, change in genetics? Maybe if we add a certain combination of drugs, what's going to happen? Can we have a given combination of drugs that is going to be synergistic? Is it going to have a bigger effect than any individually? Which ties into this next point of combinatorial therapeutics. So this is sort of a traditional model. This is a very simplified model of drug action. What you've got is you've got one target of a drug, and you want to wipe out about 90% of its activity. So you add the drug in, and it's shutting down this at 90%, and that cascades down, and proportionally, you wipe out 90% of these end targets, which is what you're interested in. So in theory, what you might be able to do is, if you understand the system well, you could say, okay, well, this works, but it actually has some pretty serious side effects that we want to avoid. So if you can work at a much lower level of that drug, it would be very beneficial. So maybe we could shut it down 50% of the activity, which would use quite a bit less of that particular drug, and then hit some other pathways a little bit farther down. So what you can get is, by using 3 drugs at a much lower level, that same net effect that you're looking for. So another aspect of this, which is probably, for a number of reasons, where medicine is going to tend more and more towards, development of resistance in pathogens now is becoming a huge problem. So combinatorial therapeutics is going to become more and more important. You've got a given bug that can resist this given drug, but cannot resist a combination of all 3. I know that they use this for tuberculosis. If you are exposed to tuberculosis, they will give you 3 drugs, with the idea that they're very concerned about it developing resistance. If it develops resistance, we have a huge problem. They have, and some strains are resistant to individual ones, but none so far are resistant to all 3. So they use this sort of approach. This is already happening to a certain degree. Okay, so just in closing, bioinformatics technology, lots of potential application in diagnostics and treatment. They're not well enough established, they're not quite proven yet, but the potential is definitely there. They're still relatively early phase of development, much like drug trials probably take 6 to 10 years for a drug to go from initial testing to market. Something like this is probably going to be similar. Right now it's not practical. 5 years from now it might be. Also bioinformatics technologies, because you are doing a little bit of a fishing excursion, you don't know quite what you're looking for, you're trying to identify everything within this given system, what you might be able to do is catch some novel drug targets with that fishing. So you might be able to come up with a gene or a protein. Function isn't known now, but you can say, okay, it's very much involved in this particular disease state or this particular tumor type. And then the last one, even though there is definitely some promise, is this going to outweigh the costs and required expertise? And that remains to be seen. Costs are going to get lower over time. Expertise might get less as well, as you're getting computer applications that can recognize these patterns fairly readily. So we'll have to see what happens with that. And lastly, I'd like to thank collaborators, Gerardo Ferber, Bernie Dunker, and one of his graduate students, Dara Christie, Brian Ingalls, and Bernie Glick, who's involved with some of the stuff with DIG, but I haven't sent any of the stuff that he's involved with here. And members of my lab, Andrea Spires, Zhenyu Sheng, Carl White, who did this work but has now moved on to U of T, Esther Mack, Gabriel Renault, who are both master's students, Andrew, who's going to be a master's student fairly soon, and Rasmus, who's an exchange student in my lab. So thanks to all of them, and thanks to you for showing up.
We'll do is open to questions now, and what I ask you to do, Brendan, if you could repeat the questions rather than my running around with the microphone. Makes it a little bit easier. Questions, comments?
Yeah.
Is the differential equation model a good model for cell science? Do you use linear differential equations?
Well, Brian Ingalls actually does most of that, but he is using linear differential equations, yes. And like any models, it's not exactly accurate, but it's reasonably predictive in many cases. So, and that's sort of what you want. You want it as accurate as possible. There'll always be little differences.
To do your cell cycle modeling, okay, first of all, I know you have some discontinuity in your package.
Okay, yeah.
Okay, now for differential equation, rule you have to establish. Now once this rule is not, do you sort the cells established?
Oh, I think the question was, do we sort the cells prior to the cell cycle modeling? So, yeah. Well, I know the cells, before we do this analysis, the cells are synchronized, so they're all...
Okay, so you're not the same.
Right, yeah. So they're all in the same cell state. Everything is behaving, at least in theory, roughly the same. So yeah, we do that. Yeah? Yep. Mm-hmm. Okay. Yeah. The question was, you noticed the dust specks on my gel. How important is lab cleanliness? One thing that I can say is this was preliminary work. Since we've done this, we've got a laminar flow hood in the lab. We have a clean space to work in. And also, we've been refining that. Most of the gels don't look like that, just this particular one did. In part for the same reason this question came up, that data looks nicer without those. So.
The other thing is more problematic. It has to do with the various microbes that can be resistant when they are most recently hospitals and you know what the solution is in part is to cut back on the minimal use of antibiotics, plus also use more here natural best flora. Those could seem to be being under control. But in what you talk about, it's always, this drug, that drug, and so on. And we unfortunately, the pharmaceutical industry hasn't paid most of our universities, you say. And even though Professor here, genetics was decided some years ago not to teach because of that influence. The worry of natural products. I mean, when you have a natural drug, if you like, a herb, it's much more, use nature against nature. It's much more difficult for the smart microbe to figure out a way around it.
I'm not going to repeat that question. Yeah, I agree. I think North American society is in general over-medicated. Yeah, natural approaches, I think, should be more recognized than they are, and you're right. You know, pharmaceutical companies have a great deal of resources that can be applied to drug development and that sort of thing. And as university researchers, should we be doing that as well, or looking for alternate options? Yeah, I agree. I think we should be looking for alternate options. What we're doing is more basic research, so you know, we're never, or not in the near future at least, we're going to be involved in developing given drugs. Yeah.
Are cell lines ability for analysis, or should you primary cell analysis? Cell lines, or you can bring that stuff already?
Yeah. Well, for the cell cycle analysis, we're using yeast. So it's a whole organism for sure. After that, it becomes a matter of practicality. Like ideally, yeah, you want, if you're looking at human effects, you want the most representative cells you can get within practical limits. So those practical limits may be that you have to look at some sort of cell line, or if you can find a way around that, yeah, I think that would be excellent. Yeah. In terms of actual protein, I can give protein amounts. Probably about 100 micrograms protein per gel, 150. Yeah, so reasonable amount. One of the students is looking at cells in eyes, actually, and fish eyes. And one fish eye has enough protein to run, essentially. So.
Other comments or questions?
Mm-hmm.
You pointed out that for this position, you have to remark that some people might be less willing or might lose something, like giving up their information or getting tests by giving this information.
Mm-hmm.
On the other hand, if many people give up information, individual will win because of prediction of disease and things like that. So this game theoretic exchange, is it part of health informatics too?
I can try. Yeah, the question was about balancing essentially rights of an individual to his own gene expression patterns and make sure that those are not misused by whoever, by insurance companies and so forth. Yeah, there is a balance there for sure, and I think it probably will fall to governments to make sure that these things are not applied, so it's illegal to use gene expression data against somebody. In practice, whether that's possible to implement, I don't know, and that's sort of way out of my area. It's just an interesting side topic.
Just one comment. You mentioned the problem of the health system and introducing this. It's well known that people in the United States particularly want to know about themselves, and what they're seeing now is a run to the lab to get genetic analyses. And there is a tremendous fear that this is going to lead to an overuse of diagnostic technologies within laboratory medicine. It's a huge problem in the just the number of people and the cost of doing that. It's an interesting thing is that a lot of what happens there is that you generate a lot of false positives as well, so that you end up declaring people not only that you have a disease or a propensity for a disease, but you in fact don't have a disease or a propensity for it, and you're labeled as having it. Mm-hmm. And that's creating a lot of interesting areas. It's a whole new world where Canada is avoid screening, where you go to a doctor with nothing wrong and they do a zillion tests and generally find something wrong with you, which generally is not really wrong with you. It's happening all over again, except happening at a much higher level, if you know what I mean, yep. And a much more expensive level. Is much talk about this in the bioinformatics community, or?
No. Short answer is no. Yeah, I think a lot of these sociological issues are extremely important and extremely interesting. but often they don't really come into the basic research. I think, you know, as researchers, it's important to consider these things, but is it going to affect what I do on a day-to-day basis in the lab? Probably not.
Because these things eventually translate into health system services, ultimately. You know, somebody comes in, a screening is done, and then it results in the system having to respond to that. Even if there's nothing really wrong with the person, the system has to discover that, and there's a huge cost. And on this continent, more than the States than here, but we seem to have that desire to want to know these things, even if they're of no value.
Yeah, well,
nothing can be done about it.
okay. Yeah, I think, and that would fall on it falls to the responsibility of the medical practitioners to say, okay, this isn't useful. I know, actually, I know a surgeon, and his opinion on MRIs is that in general, they're not necessary. If you're a good doctor, you can do a diagnosis. You don't need this nice picture. But in the health system now, like, this is one of the big things. Everybody needs MRIs, and you know, you hurt your knee, okay, you need an MRI at that kind of thing. So yeah, I think as these technologies come up, they do have to be used responsibly, and your average guy on the street isn't going to know about those, so it falls to the health practitioners to say, okay, yeah, I think this is realistic to use this in this case, or not.
Well, you know, the other part of it, if you go in and you've got a sore knee, your a priori probability of having something wrong with your knee and an MRI finding something is much higher than if you go in to Vancouver right now and for $500 get a total body MRI. I mean, it's not a hypothesis-driven or an objectives-driven process there. It's just a raw screen of data. And that's being paid for by people. I know a friend of mine went down and had a cardiac screen. They found some calcium in his coronary arteries, and now he's all worried about it. You know, so what? But the fact is that the technology itself generates cost for the system, and if it isn't disease or symptom or hypothesis-driven, there's a reasonable probability that statistics in laboratory medicine, you do 12 tests, one of them will be abnormal. That's overall. And there's nothing wrong with you. The test is just abnormal, not you. So I think it's an interesting point with regard to the area of testing. I don't know.
It's a sort of example of scarce resources being used by people who have extra money, and so those resources are less available to others who need it more.
It could be. But even the people with extra money, you have the test done, and then you go to a doctor, you see, and the doctor says, oh my God, I better do something here. And the system ends up doing further investigation, unnecessary investigation. This is what's concerned about some of this thing hitting the health system. I think there are huge implications for bioinformatics in the health system. It's going to change the use of antibiotics. It's going to change all kinds of things. It's going to change how to keep registries. And by the way, Bill Hirsch is speaking on January, remember, around the end of January here, from Oregon Health and Sciences University. And part of his talk will be related to the problems he created in information retrieval by genetic information and how some of the solutions come forward. But being able to access, for example, patient records, not just by everybody with chest pain, but by everybody with a certain gene expression. So, just a general point. Any other comments, or we're running over it, please?
Well, just let me say, a key phrase here is social responsibility of science. You can't just be in your lab, okay, and someone else is doing something to develop. You've got to take some responsibility for that. Not easy, I agree, but
Come on.
No, I more or less agree with that.
Any other comments? If not, thank you very much. Brendan, thank you.
Okay.